This is an overview of an article entitled Diminished PRRX1 expression is associated with increased risk of atrial fibrillation and shortening of the cardiac action potential, published in the October 2017 issue of Circulation Cardiovascular Genetics. Atrial fibrillation is the most common cardiac arrhythmia and is estimated to affect more than 33 million people worldwide, with substantial morbidity and mortality. As shown in this schematic on the left, the normal heartbeat is a coordinated process where an electrical signal spreads from the SA node across the atria and then via the AV node to the ventricles. In atrial fibrillation, on the right, the atria are activating in a chaotic way due to multiple electrical signals firing at very fast rates. Most of these signals are blocked by the AV node, but more beats than usual reach the ventricles and result in a fast, irregular rhythm. Atrial fibrillation is a partly heritable disorder, and a number of genome-wide association studies have been performed to better understand the genetic factors underlying atrial fibrillation. In the GWASs design, one examines the DNA of many individuals with the diseases, the cases, and many healthy individuals, the controls. There are many common variants, single nucleotide polymorphisms, or SNPs, throughout the human genome. Most SNPs have two alleles. In a GWAS, the question is asked of each SNP in the genome: is there a difference in the relative frequency of the SNP alleles in the cases versus the controls? If there is a statistically significant difference, it suggests that the SNP is marking a region of the genome, a locus, containing some genetic factors that influence the disease process. These are the first 9 loci with SNPs found to be associated with atrial fibrillation in GWAS studies. Of note, more recent GWAS studies have discovered even more loci than are shown in this table. In the paper under discussion, the authors focused on just one of these loci, the one containing the gene PRRX1. A few things should be noted. First, prior to GWAS, the PRRX1 locus had no known connection to atrial fibrillation. Second, the size of the locus's effect on atrial fibrillation may seem quite small, a 14% change in risk per allele. However, when extended across the entire population, this locus has a substantial effect on the global incidence of the disease. Finally, GWAS SNPs are almost never found within the coding sequences of genes, but rather in non-coding portions of the genome, often far away from genes. If one wishes to fully understand a GWAS discovery, one has to answer 4 questions. What is the causal variant in the locus? In other words, which SNP is directly contributing to the disease process, with the caveat that multiple SNPs in a locus can be making contributions. Second, what is the mechanism by which the causal variant affects gene function? Third, which gene in or near the locus is the causal gene? There can often be many genes in a locus. It's also possible that more than one gene in a locus contributes to the disease. Finally, what is the mechanism by which the causal gene contributes to the disease? The authors of the paper under discussion used a combination of approaches to tackle these 4 questions for the PRRX1 locus in atrial fibrillation, as shown in this schematic. We'll go through these one by one. The authors first performed fine mapping they sequenced the full 158 kilobases of the PRRX1 locus in several hundred atrial fibrillation cases and several hundred controls. They identified more than 1,600 SNPs across all of the individuals in this cohort. They then performed association analyses with each of the SNPs in this cohort, asking whether there was a difference in allele frequency in the cases versus the controls. Effectively, this was a locus-wide association study, rather than a genome-wide association study. The hope was to find a single SNP that far and away had the strongest association with disease, suggesting that it is the causal variant. As usually happens, there was no one SNP that was clearly the winner. Within a locus, SNPs can be strongly linked to the point that they can't really be distinguished by their strength of association with the phenotype. In this graph, the x-axis is position in the locus, and the y-axis is the strength of association. The green line with the arrowhead is the position of the PRRX1 gene. Here, you can see that there is a large cluster of linked SNPs. In principle, any of these could be a causal variant. So further work is needed to figure out which one might be causal. It's worth noting that the cluster of SNPs is upstream of the gene. This suggests that the causal variant may lie in a regulatory element that affects transcription of the gene from a distance, such as an enhancer. The next step was to narrow down the list of candidate causal variants. The authors did this by using publicly available epigenetic data and other data. Without getting into the details, these various graphs have peaks that indicate regions in the locus that have transcriptional enhancer activity, conservation across species, epigenetic marks for enhancers, and DNase-1 hypersensitivity, indicating open regions of chromatin in cardiac-related tissues. By comparing these graphs, the authors were able to define 8 high-priority regions. The authors took the sequences of these regions and further analyzed them. First, they linked each of the 8 regions to a gene encoding a fluorescent protein, and then injected them into zebrafish embryos. The premise was that if any of those regions had enhancer activity in the heart, they should cause a fluorescent signal in the heart muscle of the resulting zebrafish. As you can see in this figure, 2 of the regions, labeled E and F, show strong enhancer activity in both cardiac and skeletal muscle. Having honed in on regions E and F, the authors then identified every SNP in the 2 regions, isolated smaller regions of DNA around each SNP, made 2 versions of the DNA with each allele of the SNP, and linked them to the gene encoding luciferase, which can be quantitatively measured with a chemical assay. They tested these SNPs in an atrial myocyte cell line to mimic the tissue of relevance to the disease. They found that only one SNP showed differential luciferase expression activity between the 2 alleles, the SNP RS577676. Of note, the allele that is associated with higher risk of atrial fibrillation, the risk allele, showed lower expression than the other allele, the non-risk allele. Since the luciferase experiments were performed in cells, the authors next wished to test the SNP in a live animal model. They returned to the zebrafish enhancer assay. They took the E region of DNA and made 2 versions, one with the risk allele and one with the non-risk allele of RS577676, linked them to the fluorescence gene, and then injected them into embryos. You can see in this figure that the non-risk allele, T, shows more fluorescence and thus more enhancer activity than the risk allele, C. This is the same pattern that was seen in the luciferase assays on the last slide. Thus, it appears that the risk allele of RS577676 causes decreased enhancer activity. Having shown that the SNP RS577676 modulates enhancer activity, the authors wished to determine which of the genes in the locus, and there are several, is associated with the SNP. To establish a link between the enhancer with RS577676 and the PRRX1 gene, the authors performed chromatin conformation capture, a technique that demonstrated a physical interaction between the region around the SNP and the promoter of the PRRX1 gene in cardiac fibroblasts. They then performed an expression quantitative trait locus analysis, or eQTL analysis, by comparing the RS577676 genotype against the expression levels of 5 genes in the locus in a collection of 121 human left atrial tissue samples. They found that there was a genotype-gene expression relationship for only one of the 5 genes, PRRX1. Along with the chromatin conformation capture data mentioned on the last slide, this is suggestive, though not definitive, evidence that PRRX1 is the causal gene in the locus. It's worth noting that, once again, the risk allele, C, was associated with decreased expression. With PRRX1 being the best candidate gene in the locus, the authors then assessed the effect of experimentally changing the expression of PRRX1 in zebrafish. They used 2 different morpholinos in zebrafish embryos to knock down expression of PRRX1A, a fish version of the human PRRX1 gene. In each case, they found that in the live zebrafish, the atrial action potential duration was decreased. Since decreased atrial action potential duration is considered to be a key feature of atrial fibrillation in the human heart, these data suggested a direct link between PRRX1 function and atrial fibrillation. Thus, the authors provided answers to each of the 4 posed questions. First, they established RS577676 as a likely causal variant. It should be noted that their work did not rule out other causal variants in the PRRX1 locus, however. Second, they provided evidence that RS577676 regulates PRRX1 expression in the heart. Third, they found that of all of the genes in the locus, only PRRX1 expression differed in tandem with RS577676 genotype, nominating it as the likely causal gene. Finally, the authors used model systems to establish that PRRX1 regulates cardiomyocyte action potential duration, which itself is linked to atrial fibrillation. In conclusion, the risk allele of RS577676 decreases enhancer activity and is linked to lower PRRX1 expression in human left atria. Knockdown of PRRX1 in zebrafish reduces action potential duration, a hallmark of atrial fibrillation. Establishing PRRX1 to be a causal gene for atrial fibrillation suggests possible strategies by which to treat the disease, such as increasing the expression of the gene in the atrial tissues. Finally, the combination of genetic and functional approaches used by the authors in this study defined a novel causal pathway in atrial fibrillation, and such approaches could be broadly useful in the study of many other diseases, cardiovascular or otherwise.